Hello, this is a short video for those who are using Wisconsin Fast Plants in the classroom and need some data visualization tools to visualize the data that they're collecting. So for example, you might have in your class students working individually or in small groups, and they want to investigate the impact of an environmental variable. So temperature, light intensity, light color, nutrient availability, population density, soil volume, and many others that the students can do on their own experiments and then measure specific plant phenotypes or plant characteristics under these different treatment conditions. So you are performing your experiments and now you're collecting your data. And today I'm gonna just briefly talk about what is the best practice to writing down these data sets and how can I visualize it. So the first thing that we want to, when we are measuring the plant phenotypes, is we want to write that down in a spreadsheet. And there could be people that it seems intuitive to have different spreadsheets or different tables for different treatments. So it could be that you're measuring the length of, if you have two treatments, let's just say light and dark, if you're investigating the effect of light intensity, you will be measuring the length of the plants on the dark treatment and the length on the light treatment. And perhaps it's intuitive to you to have it in separate tables. But it turns out that this is not good practice for data science. And it is better to have just one table that has all the plants in your study. So we want a spreadsheet where the rows represents different plants, and ideally you will have one column that is the plant number, plant ID, so you're numbering your plants. And then you have different columns for your treatment and your measurements. So your treatment here, in this case, we have the dark light option, but it will depend on the specific experiments that you're running. So that treatment could change. You could have more than one treatment, whatever number of treatments. We have it here. And then we are measuring different things from the plant. It could be root length, shoot length, total length, but also number of seeds, days to flower, width of cotyledon, number of cotyledons. There are many measurements that we can record. And for each of those measurements, we will have one column in our spreadsheet. There are specific things that we want to keep in mind on this spreadsheet. At the end of the day, we want to save this file as a CSV file, which is called comma separated file. I will show how we do that in a second. We need to give a name to this file that does not have any spaces or any special characters. And the same is true for the text inside the spreadsheet. So we want meaningful column names without any special characters. So if you look at our data frame, I'm going to show you one example here. This is on Google spreadsheets, but you could be the same on Excel. You will see here the names of this column. I don't have spaces, I have underscores under them. So this is one important key for the text. We don't want to have spaces. We don't want to leave empty cells. So in this case, we have all the plants that correspond to the treatment light. And then immediately after, we have all the plants on the dark treatment. There is no space in there. So sometimes we like to add space, we have to add colors. Those make the table human readable, but not necessarily computer readable. And finally, we want to just keep one thing per cell, one measurement. If your data set is really big, you might want to create a data dictionary, which basically just is listing each column name at what exactly it is measuring. And lastly, you don't want to do any calculations on the data files. You know that sometimes Excel allows you to calculate things. We don't want that. We only want the plain numbers, and just don't want to use font or colors to highlight data. So we can do that ourselves to make it easy to distinguish light versus dark. But as long as we always keep that column that has the treatment. So the color should not be the separating, just light, yellow, dark, gray. Then the computer won't be able to detect that. We need to have a special column that represents that treatment. And again, these practices are not meant to make the table human readable, but computer readable. And if people are interested to read more, there is this paper here that I can link later. So now let's say that we have our spreadsheet. We have collected each row, like I said, corresponds to a plant. We write down which is the treatment for this plant, and then the measurements that we have. We can have as many columns as measurements from this specific plant. And then we can add all the different rows in there. If you have different groups of students working on different experiments, you can always merge all the data set later on, as long as they are all using the same template with the same columns. Now, let's pretend that you already have your spreadsheet. What you have to do now is you go to the Wisconsin FastStats website, which is the one here on top. I'm going to pause for a second if people want to write it down, but you can always refer back to this video. This Wisconsin FastStats is a data science tool that we created at Wisconsin, at the University of Wisconsin-Madison. There is a summary, there's a lot of information in the website. For now, I'm only going to focus on the web apps. There are two web apps, the Kotlin and the Ecosystem web app, each corresponding to a specific webinar for Wisconsin Fast Plants. You can look at the details in here. I'm briefly just going to mention, highlighting what happens when I click on Ecosystem. You will see different tabs on the left. Data upload allows you to upload the file that you want to use. Here, there are different options. So you can either browse. If I click on browse, I will try to find my file, or I can simply just drag and drop into this window, and the data appears. There is the header option. That means that my data has column names, and they are separated by commas. And one thing that I did not mention here was that if I want to download this file, there is the option of comma separated values. This is the option that we want to use, and it's the same for Excel. So they're separated by files, and the web app will show only some rows, but I can click on all, and then I can see all my data in here. Now, after I've uploaded the file, I go to data visualization and I pick the type of plot that I want. Currently, we only have 3 types of plots. The box plot is one of the most famous ones. There is a short description here. We need to pick the variables that we want to plot. Usually we want to plot the treatment variable because we want to see differences in treatment, and then one of the responses. So let's say total length. And there are different options here that I will highlight later, but if I click box plot at the bottom, I see the plot. So here, each of these points corresponds to a plant. And if I'm on top of this point, I will see the actual values. So X corresponds to the treatment. That's the X axis, that's dark. And the Y axis is the total length. I chose these variables here. And then there is a box plot that represents the mean. I will show that the mean, the median, the maximum. So here, we're giving different options to, well, first of all, add the points or not add the points. If I don't add the points, then I only see the trends. So for example, here I only see the median, the quartiles, minimum and maximum of each of the treatments. If I click on add the points, then I see the actual plant points. I can change the color of my plot, and I can change the shape of the point from circle to square. And then I can show, change the transparency of the point. If you see there's a little transparency, I can change that to being more solid color or more transparent. And the point size. So for example, this is a point size of 2. I can make it bigger, the point, or smaller. I can play with this tool. If you like this plot, you can download it. Here, there is the download option on this camera, and then you will just download this plot. And just pointing at some help, there are frequently asked questions. There are different options. If you find errors, if you run into problems, if you have feedback that you want to give us, all the questions will be answered here. So thank you for listening, and I hope that you will use Wisconsin FastStats and Wisconsin Fast Plants for your class. Thank you.